first speaker is Mark Davis, who doesn't need an introduction, therefore just a brief introduction. He's the director of the Stanford Center for Human Systems Immunology. He was for many, many years the director of the ITI, and he is, of course, well known for large and groundbreaking studies in T-cell immunology and systems immunology in many immunologically important disorders or conditions, including infection and vaccination. Mark, please.
All right. Since you took away the microphone, I'll use this one. Yeah, I want to start, since it's a vaccine symposium, it's important to start off with vaccines. And I think what I want to emphasize here is this compilation of some of the failures. You mostly hear about successes, and there have been some fantastic successes over many years now, and you just heard about some of them, especially COVID, but there are a lot of failures too, and repeated failures in HIV, tuberculosis, malaria. So there's kind of a vaccine, but it's not the greatest vaccine, I would say. There's actually the dengue vaccine made it worse. And I think the influenza vaccine has been terrible. Even with some of the latest technologies that you've heard about, it's only ever been about 60% efficacious. And so I'll talk about that, because I think we cracked that particular problem. But what also struck me when thinking about vaccine failures is we don't even know why they failed. And partly because the tools for understanding human immune responses were so primitive 20 years ago. You look at see what was available in the mouse. Immunology was going ahead by leaps and bounds, and human immunology was going nowhere. But you can tell why, because if you looked at the technology people were using, most of you couldn't use any of the mouse technology, and nobody was developing new technologies that were particularly suited towards humans. And so that's why, as part of my tenure as in directing ITI, I really thought there's a huge deficit here in the technology for understanding human responses. And also had to be built around blood, because blood is what you can get. And no one works on blood in mouse immunology, because there's so little blood. Why would you work on blood? You can get spleens and lymph nodes. So anyway, I think the essence of a good strategy is to maximize the advantages of a particular system and to minimize the disadvantages. And that whole equation is different with humans than mice. And so you basically have to work with what you can get. And we did, and we found NIH was particularly receptive to funding us and helping bring in the technology pieces and so forth. So I think we ended up with really quite an impressive collection of technologies that mostly work with blood, that can help us understand different immune responses. And as Bali said, it was also clear that vaccines were a great way to stimulate human immune responses safely. Approved vaccines, not to develop new vaccines. I think that's something best left to companies in many cases, except of course the Oxford group. Anyway, I think it worked out really well that we got a whole suite of technologies that help us understand vaccine responses, and particularly what I think one of the more recent advances has been immune organoids. Is that we found that people were whacking tonsils out of innocent little children just next door in the hospital, and that it was just full of lymphocytes waiting to make responses to vaccines. And Lisa Wagger took on this project and really developed a very simple system whereby you just make a single cell suspension of tonsils, and tonsils have like over 1 billion lymphocytes, and she found you could culture them and you could simulate them with flu and other vaccines, and they produce germinal centers and dark zones and light zones. You get antibodies, high affinity antibodies after a week or so, and T cells as well that are specific. And we also were able to show that actually they don't just make germinal centers, but they actually affinity select. That as you put in a flu antigen, it will select for higher affinity B cells, and in terms of affinity maturation and the accompanying somatic mutation. So it does a lot of the things you would hope it would do in terms of adaptive immunity. And we've been running with it ever since, and particularly using it to address some of the classical problems in flu vaccine, especially. And one of the thing that has really been holding back the flu vaccine efficacy has been this problem of strain bias, and that is that there are major strains, H1, H3, and B, that most people have a particular preference for one of those three, but not all three. And so even you get a vaccine that has 3 or 4 strains, which is the standard, most people will only make a good antibody response to one of those strains. And that's where the limit to efficacy has been mostly, because of course you may really like one strain, but you might get infected by another strain, and then you'll get sick because you don't have as good an immune response. So that's the dilemma. So people, of course, were trying to think why this was true. And this is where this very influential paper of Tom Francis came out in 1960 with an almost biblical pronouncement that which he called the doctrine of original antigenic sin. And the hypothesis was that this strain preference is due to the first influenza strain that you were infected by. And this has had a stranglehold on the field for over 60 years. And because there's nothing you can do. So, if it's all about the first strain you're infected with, what can you do? Nothing. So people did nothing. They didn't even, but worse, they didn't even test this in humans. There was no, as far as I can tell, there was no critical test of this hypothesis in humans. So, so this guy comes along, Vamsi Malajosala, fantastic postdoc in my group, who did his PhD thesis on flu. And he actually challenged this dogma, which most people just accept. And this is a good lesson in how you should not accept dogma, especially if there's no real test of that in humans. There are lots of tests in mice, but there are reasons actually why the mouse tests are not relevant to humans. So, so Vamsi did 2 tests in human cohorts. One is we have a really nice twin cohort at Stanford, and he looked at 78 monozygotic twins from the cohort, and found that in most cases the twins had the same strain preference. 90% had the same strain preference as one twin versus the other. That was good. So it says, but you can't separate genetics from environment in that case because they grow up together. We have found no twins that are separated at birth, which is really very sad because it could have been some interesting stuff there. But what was striking was actually when you look at cross-reactivity of the twins, they were not similar to each other. Looking at a whole range of different strains. So that brought into question whether, is it really, is this a real original sin? Is this really something? And then I think what was really decisive was we collaborated with Mary Stott at Cincinnati Children's, where she has this amazing birth cohorts where every week from birth, the mothers swab the noses of the kids and sends it into the lab to be tested for flu. And so she has a whole bunch of kids, infants that have never been infected with flu. And but then they're vaccinated at 6 months or a year. And we analyzed, VAMC analyzed 15 of these kids that had never gotten flu. And they all had, almost all had a strain bias based on the vaccine response. So that was, I think, kind of decisive to say, you know, what is this thing that people believe? It doesn't seem to really be going on here. And so what were we left with? We were left with, okay, there's clearly a genetic effect, a genetic predisposition. So where is that coming from? And for this, VAMC went back to sort of the basic immunology of T cell B cell cooperation. And as most of you know, a particular B cell would have an antibody against a flu antigen. It would internalize that antigen, make peptides, present those peptides to T cells, and get T cell help, which is critical for most vigorous antibody responses. And then the weak link of this is actually class 2 MHC polymorphism, which there's a lot of in humans, although not hardly in mice, which is why the mouse experiments are kind of not relevant. And so, and then you go to the immune epitope database and you plug in different influenza antigen sequences. And what the database will tell you is how many peptides could be efficiently processed and presented for a particular MHC. And what you see with the 3 main strain types is there's a huge variation that these class 2 MHC alleles, like the H3 allele, that said, they hate the B allele. So they're going to make a good response to H3 and a lousy response to B in terms of numbers of peptides. And then you have other alleles which are the reverse. They really like the B strain and hate the H3 strain. And he was able to get corroborating evidence with peptide pools and showing these predictions actually corresponded to CD4 T cell responses. But the killer experiment was this one using tonsil organoids, where if you make a vaccine, a flu vaccine, the way flu vaccines are normally made, where you grow up each strain separately and then mix them together for the, 4-strain cocktail, versus a situation where you took the same vector and mixed all the different flu types. And what this does is it allows every B cell specific for any flu strain, even the least favored ones, to pull in antigens from all the strains, including the more popular ones. And so it's going to get the maximum T cell help because it's able to get flu T cell help from any of these strains. And this works. So basically, IIV is the standard seasonal flu vaccine with 4 strains. In this particular tonsil, you can see the H3 gives you the best antibody response. The other strains give pretty poor responses. The cocktail basically mimics that IIV result, but it's where you combine all the strains into one vector that now you can equalize the antibody response almost perfectly. So even, again, even the least favored B cells in that particular person you can boost up that antibody response. And then to be even push this a little further is you might think, it was mentioned about bird flu as being possibly the next pandemic. You might think that you have seen a lot of flu and you might have a good response to the bird flu, and you'd be wrong, because if you look at a typical set of tonsils with the bird flu hemagglutinin as a stimulant, that's pretty crappy. It's barely above background in terms of antibody responses. But if you make a construct that incorporates not just avian flu as the main determinant to target those cells, but includes some of the other circulating strains so that you get T cell help across the board, Now you can boost up that antibody response really substantially. So I think this has done two things. One is I think is a substantial knock on this original antigenic sin hypothesis. And I would note that, you know, religious institutions have been trying for thousands of years to abolish sin, and with no obvious success. I don't know if anyone here is, you know. So anyway, that's just a joke. So we've also taken another approach to vaccine and antigen discovery and vaccines with a very different approach than is classic. That is, classically, you're looking for immunogenic antigens for a particular pathogen. And if you know the receptor, then you're looking for the receptor antigens that will block internalization. But you don't always have that option, nor does it always work. So we took a different approach, and this came from a graduate student, Jake Glenville, who was part of the computational systems immunology, one of the first class of that at Stanford. And he had this crazy idea that if he just made tetramers to different common viral antigens and then isolated T cells from people in the blood bank donors and looked at the sequences, he might see some useful clues as to specificity. And we always knew the CDR3s are the main determinant of antigen recognition. But until he pulled out these sequences, we hadn't realized that within the diverse CDR3 sequences, which are binding the peptide, there are motifs of 3 or 4 amino acids that, when you look at structures, are actually the contact regions of the T cell receptor CDR3 on the peptide. So they're conserved because that's the business end of the CDR3. And that was true for both alpha and beta chains in this study. And being a computational guy, he wrote a program called glyph for grouping of lymphocyte interactions by territorial hotspots that allowed you to look for these motifs and then cluster T cells that had them together. Even there might be other sequences in the CDR3s that were quite different, which was part of why it was so confusing for a long time. And so the first case of this came by looking at almost 6,000 sequences in a TB cohort that a postdoc in the lab, Hong Kong, had curated, done single cell analysis and sequencing of. And it was really eye-opener that we're able to find clusters in just this really early cohort of 20-some people in the TB endemic area around Cape Town, South Africa, that this was really working, that you can see clusters of sequences from different individuals that had these motifs, and that these also correlate with particular class 2 MHC alleles. And, you know, class 2 MHC alleles in humans generally are just nightmarishly divergent, and in Africa, they're even more divergent than that. So we already knew this was working because we got half of the specificity. And in fact, these all did correlate with TB peptides. Every cluster here recognizes a different peptide and uses a different class 2 MHC. So then this looked very good as a way to try to understand T cell responses across different people. And I should mention that you need this because looking for sequence identity doesn't work. That even monozygotic twins have very few identical T cell receptor or B cell receptor sequences, because in both cases they're so diverse that and there are many sequence solutions to the same peptide MHC or antigen. So anyway, a long story short is Chunling Wang came into the picture, wrote a much faster version of glyph, incorporated some ideas that come out of another effort at this from Paul Thomas called TCR DIST. So you're looking not just for motifs, but also for similarities, statistical similarities in CDR3 regions. And more importantly, this could now do 10 million sequences in an analysis. So now you could really ramp things up and get into big data sets. And it's easy to get millions of T cell receptor sequences from blood samples in a cohort. But the whole approach allows you now to compare people with each other and people that are having a good clinical response versus people that are having a bad clinical response. And this first came to a head in a study with Tom Scriba's group in Cape Town, where we looked at, he had 44 individuals in this context of this actually famous study, a huge cohort that had 44 individuals out of 6,000 who progressed to active TB disease in the 2 years of the cohort. And we compared that with a similar number of people who controlled the disease, who did not progress, and looked at tens of thousands of T cell receptor sequences. And what we saw is that when we did glyph analysis on that, something like 3,000 glyph groups that came out, almost all of those groups were shared between the progressors and the controllers. Only a small fraction were actually correlated with control or progression, as shown here. So that says that immunogenicity is necessary but not sufficient for protection. That there's a lot of shared T cell sequences and specificities that don't seem to matter. You can argue that, well, if you have hundreds of them, maybe they'll matter. But just in the sort of pure thinking about it, first glance, this says they don't matter, and that kind of makes sense, because if you have a pathogen with a large genome, why not devote a lot of that genome to stirring up the immune response, of simulating irrelevant T cell responses and B cell responses for that matter as well. So yeah, okay, thanks. That's my rating. It's gone down from 5 to 3. So and there's more to the story. We've looked also at people who resist TB seroconversion, and Fei Gao has done this, and we basically see that even people that are negative for TB tests, like the quantiferin assay, actually have TB-like responses, and some very dramatic. There's a particular antigen called the RV2140C that is huge in quantiferin negative people, like 1% of the CD4 cells, and then this disappears if they become quantiferin positive, or from what we can tell. So it seems like there's evidence here, and I won't have time to go into it, that the TB actually changes your T cell response with maybe repeated exposure. But just going back to the whole immunogenicity versus the significance thing, John Hausman and Chunling, sponsored by, and also with Pras Jagannathan, looking at cohorts of people that are resistant to malaria versus people that are susceptible. We did basically similar TCR analysis, and we're still in the middle of this, but the long story short is we're finding the same thing as in TB, that only a small fraction of the glyph clusters correlate with protection. Most are shared between people that are protected versus people that are susceptible to malaria. So I think, again, that immunogenicity is necessary but not sufficient. And now we have this second example of this. And then lastly, we want to move these organoid models to be more relevant to vaccine responses. And so we worked with the Donor Network West, because they collect and distribute organs from across Northern California, and hundreds a year. And especially we started with spleens. Spleens are thrown away in this, they're collected, but then they're thrown away because no one wants to vaccinate a spleen. No one wants to transplant a spleen. Spleens are more diverse than tonsils, so there's a lot of advantages. We can see some cases yellow fever response, which we never could see with the tonsils. So de novo responses are better. We see things like measles, specific B cells in these spleens of people who haven't seen measles in 50 years. So, and we see a diversity of immune responses and phenotypes, and I think that's going to be very interesting. And so we've been collecting and banking over 100 spleens from organ donors, and we haven't stopped. And that's, I think there's going to be a lot of value there for lots of studies. But the last one I want to, since I only have 1 minute left, I'll leave you with is that our dream has been to combine tissue organoids that are infected with spleen organoids from the same donor. And so we've done this working with Calvin Kuo. He's developed a very innovative lung organoid system where you can take lung biopsies and culture them over weeks and weeks. And this is a study they're just submitting where they infected the lung organoids with SARS-CoV-2, and they got some really interesting responses. And in my group, Azim Mozin has been making lung organoids from the spleen donors as well as spleens, and then putting them together where you basically you co-culture them where you have lung and spleen organoids in the same well, and then you infect the lung with flu. And we see the mobilization of dendritic cells going from the infected lung to the spleen cells and stimulating antibody and T cell responses from that. So, we have tested, so we're also monitoring the status of the flu. And we can see that over time, the spleen suppresses the flu. And especially if you prime the spleen with a flu vaccine just before you do the infection and co-culture, you shut down the flu virus in no time at all. So just to sum up here, a major problem in human immunology has been how could we do mechanistic experiments, especially if we think there's a human-specific immune mechanism, which I think a lot of evidence points to. We need a human system. Mice are not going to tell us about human-specific mechanisms. So organoids are a way to do this. We can knock out genes. I didn't mention, but we have papers and we knock out genes very easily. And then I mentioned that original antigenic sin hypothesis and evidence against that, and that we actually can make better flu vaccines if we spread around, if we borrow T cell help from the less favored strains to help the, borrow T cell help from the most favored strains to help the least favored strains. I talked about spleens. I talked about combining spleens with lung. So to thank all the people that made this possible and lots more as well. Talked about phase work, VAMC. Elsa Sola has been our organoid champion. I mentioned the work with malaria, John Hausman, Azim Ozim, and others. And particularly Gates Foundation, which dragged us kicking and screaming into the TB and malaria worlds, which might not have otherwise had the courage to go to. So I'll stop there. Thank you.
Any questions?
Mark, I'm curious if you've ever repeated those multivalent antigens with the spleens, just because there's more and or even B cell depleted them, because where they have more myeloid cells, whether that might actually change the degree of, because your model is really that it's the B cells presenting antigen, but what if it was the macrophages or the dendritic cells that were presenting the antigens? Would you lose some of that strain specificity and that ability to cross prime?
No, I think that's a good point. We haven't tried to do that. We will, because in the context of the flu infection, we're definitely going to try this with the spleens, and so we'll see if we see anything different.
Maybe just a related question. So when you do this vaccine with the multiple spikes, HA, is this in what? Is it liposome? How do you how do you make it? I mean, is it
Yeah.
liposomes? What are
So we're not clever enough to do something like they can do with Seattle. So to design something from scratch. So VAMC just found a bacteria that has a self-assembling 6-dimer, and then put a vaccination site on that. So we have a 6-dimer nanoparticle.
But maybe I'm building on that. So you would expect to see the same with mRNA vaccines, right? Where you express all the antigens on the same cells.
Right. No, I think, I mean, as Catherine is pointing out, this is a very B-cell centric thing. And in the tonsils, actually, we eliminated dendritic cells and it completely shuts down everything, but we can restore that with the type 1 interferon treatment. So the dendritic cells are, at least in the tonsil system, are not a main driver of antigen presentation.
You showed in your last slide the lung combination of lymphoid organoid and lung organoid. Can that be done with other tissue as well? And then
Yeah, I mean, I think that's the advantage of working with the organ transplant. So is that most things are really pretty available. You just have to ask nicely. Do you want skin? You want gut, bone marrow? These things, especially if they're not being used for transplant. Now, lungs are used for transplant, but they reject a lot of lungs. So they have very strict criteria, and so a lot of lungs are not used for transplant, and we get those.
So one question we have as clinicians is when to give the booster doses. Can you use your organoid system to develop a method to test serum for residual activity against the virus you're testing?
Well, I mean, I think in the context of what Bali talked about in terms of priming and
I mean, what level of antibody in a patient is still sufficient to prevent infection? And can you test that?
Well, I mean, that's testable. Yeah, we haven't done that.
That'd be an interesting thing to do, yeah. Clinically relevant.
All right. Yeah.
You showed that the TCRs are not the only thing to
Can
determine.
you speak up?
You showed that the TCRs are not the only thing to determine the vaccine. Could the phenotype of TCR plus TCR sequence, could that affect the candidate selection for vaccination?
I can't really hear the question.
Oh, how I think that, can you hear me? You said that TCRs are like not the only thing to select vaccination. Does the phenotype of TCR, could that help in filtering out the redundant candidates?
Is this in the TB response?
Yes.
Yeah, actually, that's an interesting thing I forgot to mention is that these CD4 T cells that are coming up in people that are quantiferin negative actually have a follicular helper cell phenotype predominantly. They don't have the Th1 star. So something really interesting, and the follicular helper phenotype has been associated with protection in some mouse models. But yeah, that's another striking thing that seems to happen, and I think it's happening with repeated exposure. We know that in South African cohort that at age 10, about half of the kids are quantiferin positive, but by the time they're adults, 70–80% are quantiferin positive. So there's a real shift there towards this quantiferin positive phenotype, and then you're changing this T cell response dramatically. So it really looks like a case of a pathogen kind of shaping your immune system to better suit its needs. May not be your needs, but it's the pathogens. Yeah, Polly.
I shouldn't be asking a question because I mean, I could ask, but it's fascinating because everyone believes that there are these long-lived bone marrow plasma cells and that the bone marrow is the major compartment for these cells. But in humans, nobody's really carefully looked at spleen, tonsils, lymphoid organs, other organs. And it sounds like you may have an opportunity to actually, from the same person,
Yeah.
do a balance sheet of how many measles-specific plasma cells can you isolate from bone marrow, from spleen, and so on and so forth. I suspect that there will be many other sites where these plasma cells live.
And also there's some advantages in terms of the spleen culture. When we're using spleens alone, the plasma cells have nowhere to go. So they stay there. So we actually see them. And so it might be an advantage to have them not being able to go anywhere. But then of course you can engineer them to go, again, with this sort of multi-tissue culture system, definitely that would be something you could look at. Yeah. All right.
Thank you.